Hello and welcome to Math Simplified. In this video, we will discuss about the reverse transcriptase polymerase chain reaction, also known as RT-PCR. To understand RT-PCR, first you should know about the polymerase chain reaction or PCR, which I have explained here in this video on our channel. So pause this video right here and take a look at the PCR video first. Links are in the description bar down below. So let's begin with this video. Our world is struggling today. Thousands have died, millions infected, billions in fear. COVID-19 have brought the world to its knees. As humans collectively work to fight off this pandemic, all hope is not lost. Years of research and hard work by brilliant minds worldwide have provided humanity with the hope of survival. Drugs are being researched, vaccines being designed, dedicated centers developed, all in an effort to minimize further casualties. Our most dependable support has been lab testing of millions of samples to trace the infected and further initiate interventions. You might have heard that diagnosis of COVID-19 infection is being made within 8 to 10 hours with very high accuracy of results. All this is possible because of technique, although not new, the whole world has now understood its importance. It is the RT-PCR. Now for those of you who haven't seen the video on PCR, let me simplify it for you. Living cells have genetic material. The genetic material is a set of instructions for how the cell needs to work, what functions it will perform, and how it will grow and eventually how it will die. The genetic material can be found in two forms: ribose nucleic acids, RNA, and the deoxyribose nucleic acid or the DNA. DNA is a two-stranded molecule that is found in all organisms such as animals, plants, and some viruses. It contains the genetic code. The genetic code is a blueprint of that organism. RNA is generally a one-stranded molecule and does some different functions as compared to the DNA. The PCR or the polymerase chain reaction is a method widely used in molecular biology to rapidly make millions to billions of copies of a DNA sample. This allows scientists to take a very small sample of DNA and amplify it to a large enough amount to study it in detail. PCR was invented in 1983 by the American biochemist Kerry Mullis. Now some viruses such as the coronavirus can only contain RNA. They rely on infiltrating healthy cells to multiply and survive. Once inside the cell, the coronavirus uses RNA to take control of the cell and make it into a virus-making factory, which produces more cells which are infected with the viral RNA, and the cycle goes on. Well, this presents a problem for the traditional PCR because in the traditional PCR only DNA can be detected or copied. In order for a virus like the coronavirus to be detected by the PCR, scientists need to convert the RNA to DNA. This is a process called reverse transcription. If you remember the process of copying of DNA to RNA, which was known as transcription, hence the process of using RNA to form DNA is known as reverse transcription. Hence, using this technique of reverse transcription with the technique of polymerase chain reaction is known as reverse transcriptase PCR or RT-PCR. An enzyme is used by scientists which converts the RNA into DNA, and the PCR makes millions of copies of this viral DNA in no time. And subsequently, scientists have a large enough quantity of viral DNA to accurately confirm that the virus is present in the sample or not. A sample is collected from parts of the body where the coronavirus gathers, such as the person's nose or throat. The sample is treated with several chemical solutions that remove substances such as proteins and fats and extracts only the RNA present in the sample. This extracted RNA is a mixture of a person's own genetic material and, if present, the coronavirus's RNA. The RNA is reverse transcribed to DNA using a specific enzyme, and from here onwards the process is similar to that of a PCR. So, our goal here becomes that the minute quantity of genetic material that we have extracted from the cell should be amplified, which can be done by making several identical copies of the same by using a technique known as PCR. The first step is to add specific short fragments of DNA to the sample. These fragments attach themselves to unique targeted sections of the viral DNA if the virus is present in the sample. Additionally, these specific DNA fragments also contain marker labels which are then used to detect the virus. The mixture is then placed in the RT-PCR machine. The machine cycles through temperatures that heat and cool the mixture to trigger specific chemical reactions that create new identical copies of the targeted sections of viral DNA. The cycle repeats over and over to continue copying the targeted sections of viral DNA. Each cycle doubles the previous amount. 2 copies become 4, 4 copies become 8, and so on. The growth becomes exponential. A standard real-time PCR setup usually goes through 35 cycles, which means that by the end of the process, around 35 billion new copies of the sections of viral DNA are created from each strand of the virus present in the sample. As new copies of the viral DNA sections are made, the marker labels release a fluorescent dye, a signal which is measured by the machine's computer and presented in real time on the screen. The computer tracks the intensity of the fluorescence in the sample after each cycle. When the amount goes over a certain level of fluorescence, the presence of the virus is confirmed. Scientists also monitor how many cycles it takes to reach this level in order to estimate the severity of the infection. The fewer the cycles, the more severe the viral infection is. The fewer the cycles, the more severe the viral infection is. The real-time PCR technique is a highly sensitive and specific technique and can deliver a reliable diagnosis as fast as 3 hours, though usually laboratories take on an average between 6 to 8 hours. Compared to other available virus isolation methods, real-time PCR is significantly faster and has lower potential for contamination or errors, as the entire process can be done within a closed tube. It continues to be the most accurate method available for detection of coronavirus yet. So this was all about the reverse transcriptase PCR. I hope you liked the video. If you did, make sure to hit the like button below, and if you want access to all the downloadable content from this video and all of our other previous videos, you can join us here on YouTube, or you can also join our community on patreon.com. Thank you so much for watching.